Good afternoon, and thank you for the invitation. So I have nothing to disclose. So our model of interstitial cystitis bladder pain syndrome is based on the expression of Claudin-2. So Claudin-2 is a tight junction-associated protein that forms paracellular pores and is dramatically upregulated in bladder biopsies from IC-BPS patients. So we previously showed the adenoviral mediated expression of Claudin-2 in the urothelium resembles the cardinal features of IC, and rats that are transfused with this adenovirus have increased urothelial permeability. They present inflammation in the bladder mucosa and laminopropia, and they have increased frequency and pelvic allodynia. So they have pelvic pain. So also in addition, we show that these functional changes result from an increase in the excitability of the bladder sensory neurons with heterotoxin-sensitive action potentials. I don't know if the laser works. Okay. Sorry. So in the right panel here, you can see the number of spikes in response to electrical stimulation is significantly higher in the tetrodotoxin-sensitive neurons from rat transduced with Claudin-2 than the CFP transduced counterparts. So the goal of this project was to determine whether the hyperexcitability seen in the tetrodotoxin-sensitive neurons from rats with increased urothelial permeability reflects changes in the activity of voltage-gated sodium and potassium channels. And to do that, what we did is we performed single-cell quantitative PCR with sensory neurons isolated from rats transduced with CFP or Claudin-2. And what we saw is an increase in the expression of KB 2.2 KB 9.1. So KB 2.2 is a voltage-gated potassium channel, and KB 9.1 is an accessory subunit that associates with KB 2.2. To determine whether this increase in mRNA expression correlates with an increase in the activity of these channels, we use a toxin from a spider that is called 1-toxin-1E. That specifically blocks KB2 channels. So we use the patch clamp technique, and we measure the toxin-sensitive current in the sensory neurons from rats transduced with CFP or Claudin-2. And we saw a larger, so as shown in the panel D, the toxin-sensitive currents are significantly larger in the sensory neurons from rats transduced with Claudin-2 than the controls. So as mentioned before, the expression of Claudin-2 in the urothelial promotes repetitive firing in the bladder sensory neurons with heterotoxin-sensitive action potentials. And what we found is that the 1-toxin-1E blocks or inhibits this repetitive firing, and that is shown in panel B. And the spikes as a function of the stimulus are shown in panel C. So in addition, we found that the activity of the sodium channel, voltage-gated sodium channels that are sensitive to tetrodotoxins also are significantly increased in the sensory neurons from rat transduced with Claudin-2 compared with those transduced with CFP. So in summary, our data showed that urothelial barrier dysfunction increased the expression and activity of KB 2.2 and the activity of tetrodotoxin-sensitive voltage-gated sodium channel in a population of bladder sensory neurons with tetrodotoxin-sensitive action potentials. So the increase in KB 2.2 activity promotes repetitive firing, and we hypothesize that this likely contributes to the changes in bladder function and pelvic sensitivity seen in the rats with increased urothelial permeability. So these findings reveal novel potential targets that can be used to treat hypersensitive bladder disorders that are caused for increasing urothelial permeability. Thank you, and I'm open to questions.
Questions from the audience, please.
Thank you for nice presentation. Yoshimura from the University of Pittsburgh. So, increase of potassium channel, general concept. Increase of potassium channel usually suppress the bladder neuronal excitability. But this case, increase of that potassium channel actually decrease that excitability.
Yeah,
so how do you connect that functional properties?
Well, actually, so previous studies in other populations of the neurons show that these channels, basically, if you inhibit Kb2.2 or Kb2.1, you basically prevent the repetitive firing. And that was shown in trapezoidal neurons and other different neurons from different areas of the brain. So this potassium is important. Basically, the function of this Kb2.2 is to it's important for the repolarization of the membrane.
Okay.
And when you block it, for some reason the polarization is faster, so that the neuron cannot refire.
Ready to the next firing of
exactly.
that. Okay.
Yeah, exactly.
Thank you. We've always heard about A delta fibers. Now, the C fibers, do any of these things live on the C fibers?
Okay, so all the data that show are with tetrodotoxin sensitive afferent or sensory neurons. And based on the work of Yoshimura and DeGros, these are A delta fibers. They are not C fibers. So this is different that you have when you treat animals with cyclophosphamide. And we have done those experiments, and when you use cyclophosphamide, you basically have a change in C fibers as well in the A delta. But in this case, the increase in urothelial permeability only affects these tetrodotoxin sensitive neurons.
Thank you.